of the Department of Immunology and Microbial Sciences at the Scripps Research Institute in Florida. And before joining the Scripps in 2012, he was at Harvard for many years. And I didn't know, but he may talk about that. He did his undergraduate in government, so he did something completely different before, which is always interesting. But I'm particularly interested in his talk because he's one of these people who is now combining gene therapy with a novel way of protecting cells from HIV entry. So very excited to hear his talk. AAV-expressed CD4 provides durable protection from multiple HIV challenges. Thank you.
Okay, thank you very much, and thank you for this great conference and the chance to talk and listen. So today I'm going to talk about these three things, and I think most everybody here knows what an antibody is, including that it has an antigen recognizing region and then a region, an FC region that mediates its effector function. And most of you in the HIV field have heard of CD4 and this has been used both in the laboratory but also in the clinic and tested many, many times actually for its ability to suppress HIV infection, and it always failed, but always was shown to be safe. And then the last thing I'm going to talk about here is this thing we call ECT4. E can mean many things, maybe enhanced or something, but the point is it's just ECT4 with a small CCR5 mimetic sulfapeptide attached to it. Now, the other thing, let's see if we go. The other thing we're very interested in, and I was very grateful for David Baltimore's great introduction to this entire field, is AAV, and using AAV as an alternative to a conventional HIV vaccine. And many of you know that this field was sort of pioneered by Phil Johnson, including with this very important paper in 2009. And he was able to show that AAV-expressed immunoadhesins could provide pretty good protection against a very sort of wimpy SIV MAC316 virus. And then David Baltimore actually and Alex Balash forwarded this field and really validated, I think, to some extent this field by showing that you can express full-length antibodies from an AAV vector and then protect those humanized mice from pretty strong HIV challenge. So David mentioned very nicely that AAV can stably express protective concentrations of broadly neutralizing antibodies from muscle cells for a number of years. So a rough estimate based on Phil Johnson's data now is we've probably got a half-life of these antibodies of about 3 years. So that can extend to 10, depending on where you start from. AAV itself is considered safe. It doesn't integrate, it doesn't replicate, and it doesn't provide any viral proteins express any viral proteins. So the safety issues associated with it are going to come from long-term expression of the transgene. And what that means is we need off and regulatory switches. But as I think David and Alex Balash's work showed you, and others are working on very similar things, these are on their way. So there is a problem, and the problem, I think, is that antibodies are not perfect, even so-called broadly neutralizing antibodies. The broadly neutralizing antibodies are indeed a major jump forward on the previous generation of antibodies, but they're not perfect by any stretch of the imagination. They only neutralize about 60 to 90% of all isolates. And these we should know exactly what we mean by this. We say 90%, it means that at 50 microgram per mil, we protect against half of the viruses. We suppress half of the entry. And these are under conditions that are extremely favorable to the antibody in vitro. So that's the first problem is they're simply not broad enough to cover anywhere near all of the isolates that are circulating, or all the isolates that are in the quasi-species in a given individual. Second problem is there's no fitness cost to escape. Period. The virus has learned, and for eons now, how to escape any antibody that we have ever seen or generated. And in fact, it's very good at doing so, and there's a very short pathway typically between your starting virus and an escape variant of that virus. So this is a problem. This is a problem both for the two kinds of applications we consider for AAV. One is vaccines. So you may think, okay, we get 90% of protection, pretty good, and I would agree that would be a major step forward, but consider a little bit what the implications of that are. That means 1 in 10 individuals will get infected. When that individual gets infected, he will then become an immediate concern for all his vaccinated neighbors. So this is very different from a situation where you say a conventional vaccine has 90% efficacy. The second problem is probably more significant, and that is for infected individuals. As I mentioned, the level of escape, or the number of escape variants, suggests that, as with drugs, most individuals carry within their quasi-species viruses that already escape. And we're starting to see that now with some of the human trials, that there are an unexpectedly high number of individuals that readily and rapidly escape from the suppression of the antibody. So that these concerns need to be considered. Now, the other problem with AAV is it's a big adjuvant. That's to be expected. We're putting a lot of viral particles into the arms or legs of these animals, and those viral particles carry a lot of DNA and a lot of protein, all of which are immunogenic. So they drive the natural tendency of humans to make anti-idiotypic antibodies to passively administered antibodies, for example, and they make the frequency of those much higher. So very roughly from animal studies, about 90% of the time you see anti-idiotypic antibodies, but only about 20% when passively administered. And that 20%, by the way, is a number you see in humans with, for example, fully human anti-TNF antibodies or other antibodies that are now being used clinically. There's a number of reasons, other additional reasons, besides the fact that AAV is an adjuvant. The antibodies that we use are necessarily and extensively hypermutated, which means that they're carrying a number of both B and T cell epitopes themselves. And then we're pushing a lot of antibody through the muscle. And what we have seen now is that this level of antibody creates ER stress, and that tends to drive innate immunity itself. And it's especially a problem if you have an imbalance between the heavy and light chain, or the heavy and the light chain have not gotten together. And this may be one reason why we've been a little bit more successful with a single chain immunoadhesin. And similarly, Phil Johnson had lower anti-antibody responses than most of the other reports, Gary Nabel, Ron DeRozier, and what I'm about to show you. So this is our own example of how these experiments sort of fail. So what we did was we inoculated, using AAV, pairs of antibodies, a CD4 binding site antibody, and what then is called a V3 antibody, or a glycan-dependent antibody, in combination, in 2 different combinations, either with IgG1 or IgG2 Fc domains. So the IgG1 mediates the effector function. Those are much lower or non-existent with the IG2 Fc domain. The bottom line here is that when we challenged with our lowest dose of virus, we infected 2 of our control animals and 4 of our IgG1 animals. And we came in with a double of that dose. We infected 5 of these, all of our controls, and 3 of these. So the reason for this is that the titers here were much lower, much less than the 1 microgram per mil level that we'll probably need to protect using these antibodies. And that corresponds to a very, very high, robust anti-antibody response. These kinds of observations have been seen with Ron DeRozier. They've been seen with Gary Nabel. David Baltimore showed that you have to immune suppress to get protection under these circumstances. This is not simply due to the fact that these are human or simianized forms. These are simianized forms of these antibodies. It's very likely to do with the fact that they're somatically hypermutated extensively, and AAV is a significant adjuvant. Okay, so my talk today, or my point today, is this protein that we've made, ECT4, has significant advantages over antibodies as an AAV-delivered vaccine alternative or potentially a therapeutic. It's more potent on average than any antibody, any naturally occurring antibody. It's much broader. It gets everything. It potently neutralizes everything we've thrown at that, including SIV and HIV too. It's much less immunogenic, and as I'll show you, it works much better than the antibodies I just showed you. So AAV-delivered ECT4 will protect macaques from very high intravenous doses from CHIV88, even at concentrations we think we can readily get in humans. So to start this, how we got here, let me just remind you that, that for HIV to enter cells, it first has to bind its cellular receptor CD4. And then the big thing here, the big point for this talk, is there's a conformational change that the envelope glycoprotein undergoes that then allows it to interact with CCR5. Now, one of the things we observed 1 million years ago was the amino terminus of CCR5 is modified in a very unusual, what was then thought to be unusual way, which is sulfate groups were added to the 4 tyrosines at its amino terminus. We kind of suggested in our original observation that that was a common property of chemokine receptors. It is, and that and indeed chemokine receptors require those sulfates to bind their chemokines. But for our purposes, the main issue is that this is the initial and the highest energy interaction between the envelope glycoprotein and CCR5. And I'm sort of showing you the sequence starting from the N-terminal methionine of CCR5 with these 4 critical tyrosine sulfates and this sort of epicenter of GP120 binding where most of the energy comes from. So now what I've done is I've taken the receptor binding part of the envelope glycoprotein, GP120, turned it around, and I'm showing it in a structure done by Peter Kwong a number of years ago, and we can see where these very conserved sulfatyrosine binding pockets are positioned with respect to the GP120. And we know that because that comes from a tyrosine sulfated antibody. So this was an observation we made a few years later, and I still like it even better than maybe a lot of things we've done because it tells us a way that the antibodies, another way that the antibodies create diversity. So what we showed was antibodies have an inherent propensity to add sulfatyrosines in their CDR3 regions. So it's sort of built into the diversity region, into our genomes, that on occasion, with some frequency, sulfatyrosines can be added to the CDR3 regions. So we all have tyrosine sulfated antibodies in us at about a frequency of maybe 1 in 1,000 or something of that nature. And there are now a number of tyrosine sulfated antibodies, including a couple of the ones that Dan mentioned, which were CAP256 and PG9 and PG16, a couple of the apex antibodies. But the first class of antibodies that we identified were CD4I antibodies. These are antibodies whose epitope is exposed when GP120 binds to CD4. Okay? This is the antibody 412D. And again, we were very happy to see that this antibody helped us localize where these key sulfatyrosine binding pockets were. And then another antibody from the same individual is an antibody called E51. And this antibody is special because when we took the CDR3 and made a peptide out of it, we could precipitate GP120, in contrast to a number of the CDR3s of a number of these other sulfated and non-sulfated antibodies. So what I'm trying to show you here is GP120 and the ability of this FC peptide construct to precipitate GP120. And only this antibody P51 does so. We called it CCR5 mimetic 1, or CCR5 mim 1. And the reason we think that it works is it does a pretty good job mimicking the pattern of sulfatyrosines present at the amino terminus of CCR5, but we exclude a bunch of junk, including its O-glycosylation region, some hydrophobic residues that point downward, and other things that sort of just create hydrophobicity from the original CCR5 peptide and replace them with more flexible glycines or just sort of eliminate this O-glycosylated loop. So that's CCR5-MIM1. Now, one thing you would expect with CCR5-MIM1 is that when you add CD4, it will indeed precipitate more GP120 than in the absence of CD4, and that indeed turns out to be true, as you can see here, comparing this band to this band, this band to this band, et cetera. The other point this slide makes is that these sulfopeptides bind equally well, or if not better, to X4 and R5X4 isolates than they do to R5 isolates. And I can go into the reasons for that if somebody asks. But the main point here is that CD4 enhances the binding of the peptide. So we did what was obvious, basically. We made fusions of the peptide with CD4. And we did it in several different ways. And, the bottom line here is this one worked much better than these two or than CD4. So our basic sort of pattern for I guess most of the rest of the talk is CD4 is in red, and then ECT4 is in blue. And you can see that at higher and higher concentrations of CD4 or ECT4, we get much more potent neutralization with ECT4 than CD4. And we were sort of very happy to see that among the things that we saw enhancement was SIV MAC239. And this was our first beginning inkling that this is something that's not only very potent, but exceptionally broad. So we turned to Mike Seaman and asked him to test two different, relatively large panels of viruses that are selected to be especially resistant to neutralizing antibodies. So these are so-called tier 2 and tier 3 isolates. We did it from a range of clades. We said just pick all the tier 2 and tier 3 that you can easily get your hand on. And then we did a sort of a more systematic study of all but one isolate that has ever been shown to be resistant to these two what are referred to as broadly neutralizing CD4 binding site antibodies. And this summarizes those data, and there's a sort of a number of points. So let me orient you a little bit, because this is neutralization with CD4. Low is good. And then this is our original ECT4 construct with the MIM1 peptide. And then we made a couple of other constructs for the purposes of this talk don't matter at all. They just have a point mutation either in the sulfopeptide or in CD4. We dropped the CD4 point mutation for reasons I can talk about, but we retained the point mutation in the peptide. We call that CCR5-MIM2, and that's kind of our lead construct, so to speak. And then we compared neutralization to these three CD4 binding site broadly neutralizing antibodies. And you can see a couple of things. First of all, we always get this 1 to 2 log, 1.5 to 2 log improvement over CD4, every virus we try. The second thing is that we are indeed more potently neutralizing all of these isolates, or on average certainly, than these broadly neutralizing antibodies. And indeed, we're more potent on average than every broadly neutralizing antibodies, although there are now antibodies getting close. But the real story here is up here, where we list the number of isolates that we did not neutralize efficiently. And you can see even CD4, it's indeed turns out to be fairly easy to escape from ECT4. But we've not yet found an isolate that is resistant certainly by the field's definition and by our own more stringent definition of what's potently neutralizing. And then if you look at these broadly neutralizing antibodies, this is not a fair representation. These antibodies typically neutralize about 85% with IC50s, again, of 50 microgram per mil, very, very high. So what we summarize here is that our geometric mean IC50 is about, and that's mean, not median, is 0.5 micrograms per mil based on panels that are really biased toward hard to neutralize viruses. Our worst IC50 that we saw was 1.2 microgram per mil. Our worst IC80 was 5 microgram per mil. So these are well under the concentrations that we think we can sustain very easily in vivo. And then we hit HIV2 and SIV isolates as well. One thing we want to know immediately is whether we can mediate ADCC assay. I'll just sort of jump through this quickly because our comparisons aren't that great. We compared it with a not very good antibody, IgG4-B12. But the take home is that the peptide significantly improves the ability of CD4 to neutralize, to mediate ADCC. And most importantly for our purposes, the peptide did not interfere with the ability of the Fc to mediate ADCC. We think it's better than that because it binds the trimer in multiple modes. So polyclonal sera mediates ADCC better than any monoclonal antibody, and ECT4 may approximate 2 or 3 different orientations on the envelope, as I'll show you. Why is it so good? Well, why is it so broad? This is, I think everybody who's been working in antibody knows this instinctively. There is a non-equivalence between the CD4 binding site and the CCR5 binding site, or the CCR5 binding site, and the epitopes of any antibody. The epitope of any antibody is larger. This obviously gives the virus an immediate means of escaping without a fitness cost. ECT4 only binds necessarily and functionally conserved regions. And indeed, when we try to generate escape from these viruses in vitro, what we find is that it's much harder. Indeed, we have not yet generated full escape from ECT4. So this is what we're showing is we passage these viruses for 270 days now in the presence of the IC80 or greater of each of these reagents, and then asked whether the resulting viral mixtures were sensitive or resistant to each of these reagents. So when we just passaged without inhibitor, we kind of retained our initial sensitivity of these viruses. When we passaged in the presence of the CD4 binding antibody, we indeed got ready escape from the CD4 binding site antibody. When we passaged in the presence of CD4Ig, we got escape from CD4Ig, and someday we're going to think harder about how one goes about escaping from CD4Ig while still being very efficient in infecting cells. But the point is that when we passed in front of ECT4Ig, we got escape from CD4Ig, and we shifted the curve for ECT4Ig, but we have not yet obtained an escape variant. So the conclusion here is that it's harder to escape. It's not saying that it's impossible. Our hope and our expectation is that there'll be a fitness cost when we do get escape, and where we will get escape is in animal studies in which we force the escape. Okay, so why is it so potent? Well, this is a little bit of a complicated issue. I'm going to just jump through it very quickly. We're binding 2 sites, so we're getting avident and cooperative binding between our 2 sites. And indeed, we're actually binding 3 sites because both sulfapeptides we know are engaged to each of one GP120 of the trimer, and we know at least one CD4 is engaged either to that same trimer or to another trimer. There's no enhancement. So CD4Ig is notorious for enhancing entry at low concentrations by promoting direct binding to CCR5. So we're blocking that physical association. And then it's very likely that we're promoting further irreversible changes that CD4 also does, if only by localizing CD4 to the trimer for a longer period of time. But we may also be mimicking CCR5's ability to mediate further steps in the fusion process. These are our 2 possible models. As I mentioned, we know that both sulfapeptides are engaged, and we know that a CD4 is engaged either in cis to the same trimer or in trans with the bridge, and we're just trying to work this out. If I were to guess right now, this is primarily our neutralization strategy, and this may be mediating ADCC with more more motion on the surface of the cell. So the animal studies, very quickly, we used these 2 vectors. So one wrinkle here is that we added a vector that expressed an enzyme that adds the sulfate. So all cells carry this enzyme, but when you overexpress proteins, you saturate that enzyme, and you need to add more. And we know now we do need to add more. So we added at a 4-to-1 ratio these 2 vectors. The rhesus macaques were inoculated with these typically but large amount of AAV1 particles, again at the 4-to-1 ratio. They were challenged repeatedly with escalating doses of CHIV88 until they became until only the controls became infected. And then we measured protein titers, viral loads, and immune responses to ECT4. And these are the titers. They sort of jumped around on us. That is sort of pattern that may reflect our early days with trying to measure titers, but also sort of the typical jumpiness and pattern you see with AAV-expressed proteins. They kind of settled out here to about what we call it on the order of 70 micrograms per mil or 20 micrograms per mil. This 20 micrograms per mil is a very accessible number. It would be 1/1000 of your IgG if we achieve that. So 1/1000th of your IgG. I wouldn't want to go higher, and this should be way more than enough to protect. The other thing that was very interesting in comparison to antibodies with the same IgG2 Fc domain, this was much less immunogenic than these antibodies that I showed you earlier. I won't go into the details of why we separated that. It has to do with they have different light chains and how we measure them. But when we sort of scaled this up, so we sort of zoomed in here and tried to see what we were getting, what we observed is 2 of the animals did have detectable anti-anti responses that went away at about 36 weeks. Those antibody responses did not appear to suppress the expression of the protein, and they were not correlated with the expression of the protein. So this is the protection data. So as I said, we just went in with increasing amounts of ChIV-88. We challenged these intravenously. That is typically thought to be a more stringent challenge, less physiological for most sexual transmissions. And that at 200, we converted 200 picograms, we converted 2 of our control animals. At 400, we converted our 3rd control animal. Our 4th guy was a little bit resistant. We got him at 800 picograms per mil. At this point, none of our ECT4s were infected. We got bold and kept doubling the responses as far out as more than a year out. And then none of these responses, none of these animals were converted. So 4 and 4, obviously statistically significant, and at doses that are way beyond any challenge dose you would, ever be exposed to. So that's the talk, and I just wanted to summarize for the community and for ourselves, all of ourselves, we're the community, what we've kind of really got here. So the one thing we know we've got here is a very good entry inhibitor. It's on average more potent than any neutralizing antibody, and certainly much broader than any neutralizing antibody. It appears in vivo to be more difficult to escape than any antibody. We hypothesize and we hope that the escape virus will be less fit and less transmissible. Total speculation at this point. It is also clearly less immunogenic in macaques than antibodies, and as we solve the problem of immunogenicity of antibodies, that we anticipate that these will be even less immunogenic. ECT4 will be even less immunogenic. If we combine it with AAV and we continue to live under the immune radar, it could protect humans from most potential transmission events. However, and this is by the part where I say why you should not be excited, is that the safety of this approach has not been established. And we're really, really going to need an off switch, especially as we get into high-risk uninfected individuals. Its use as a therapeutic is exciting, but still speculative. We'd like to think that it could replace some components of ART. Nonetheless, all of these things said, this general approach, I would argue, is closer to realization than any conventional vaccine effort. And then I'm not going to thank all the people on this list. I do thank them, but I just want to note Matt Gardner and Lisa Catton, who really drove this project home, the support from NIH and the hopeful support, future support from the Gates Foundation. And I'll take your questions. We don't formally know that. We have no reason to think it won't. We think it will behave like an IgG, but we don't formally know that yet, and we're doing those experiments now. That's definitely. This is all IV challenge, right? So as we note, it's stringent, but physiologic only for IV drug users and not for sexual transmission.
I was wondering, to adapt this therapeutically, are you worried if it'll select for like export-tropic viruses?
Okay, so I need to hit that every time. So counterintuitively, this so-called CCR5 mimetic works very, very well against X4 viruses. And there's a lot of hand-waved reasons I can give you. I'll give you one that I like the most, and that is when a virus evolves from R5 to X4, it goes to X4, it goes to an R5 X4 stage. And a characteristic of those viruses is that they're very good. They glom onto the N-terminus of CCR5 very strongly, while they learn to adapt to the body of CXCR4. So that's kind of the pathway by which they go. And in that pathway, they have to bind very tightly. Of course, those things are also conserved because there's a lot of structural constraints on them. And then X4 viruses also like to use heparin sulfate, and they bind in the same region. But for whatever reason, our peptides are stronger against, well, I know the reason why they're stronger against X4, which is the X4 viruses tend to be more open. Their co-receptor binding sites are more exposed, but they bind as tightly and they work better. So X4 equal to R5 for these peptides.
So Mike, do you know when you want to do this cross-feed, does it bind to MHC class molecules?
It does not bind MHC class molecules as a soluble protein. So the
Because you have an FC receptor, so cross-feed and generate.
Oh, so the argument would be you got an FC receptor, and then because you've localized it, it could bind FC, and we haven't tested that point. And I need even to go back to the original papers that show that was not true for CD4IG, and make sure they covered that point. So I don't know that. Good question.
So there seems to be this false dogma that AAV does not integrate, when actually it does integrate and not.
Well, no, that's correct, but you'd be mistaking the vector from the virus.
The vector, I mean, there's a number of studies that show vectors do actually integrate.
Okay, okay, no, I would suggest that at the frequency of DNA. So, we all know when you make a stable cell and you put enough DNA in, it will integrate. But it's lost all the mechanisms that the virus has for integrating. Okay, if you.
There have been several studies that have shown that AAV vectors actually can cause tumor genesis in mice, albeit in the liver. Do you have any concerns long term that this could potentially happen as well?
Well, I hope we get to those concerns. Yeah, I'm much less worried about that than just turning off the protein off at this point. That will be a concern for sure in a couple of years.
Okay, one more question.
So Mike, this result that you have of expressing BNAP, immunized BNAP in AAV vectors listed in ADA is very important. It's tremendous implications for therapeutics where people are. Did you express or did you inject these immunized antibodies without AAV monkeys?
Right. No, we didn't, but Gary Nabel did. So Gary Nabel with passive sees 20% anti-id, with AAV 100% anti-id. So there's no now there's more caveats that I can go into how that antibody is expressed with a particular vector construct. So we know we can tone it down a little bit just by improving the vector, but it still remains a concern because it works like an adjuvant. Yeah.
Okay, I think we need to move on. Thank you very much for this.